Hello everyone. In today's video lecture, we are going to talk about very important and little bit complicated prophase 1 of meiosis 1. Under this, we are going to emphasize more on the structural changes in the chromosome inside the nucleus and during this particular stage. So let's talk about that. As you can see, this nucleus containing the chromatin material, this is the condition of the cell before meiosis 1. It means at the end of the interphase. So after interphase, when the cell is going to enter into the prophase 1, what first change will take place? That this chromatin material start condensing to form the chromosomes. Okay, so how the change is going to occur now? As you can see, these are the chromosomes. This chromatin material is coiled, condensed to form these kinds of structure which we call chromosome. If I'll talk about human cell, so such structures are 46 in number. Okay, as we know, we are having 23 pair or 46 chromosome. So these chromosomes which are coming from father and mother, they are 46 in number in a diploid cell. And we know this thing, meiosis is going to occur in diploid cell to form the haploid daughter cells as the number of the chromosomes are going to reduce. So these chromosomes which are coming from father and mother, they start pairing, they start coming close. And this process, when they start coming close, this is called synapsis. Okay, this is called synapsis. This is a chromosome which is containing the replicated DNA within it, that I'll show when the clear visibility of the chromatids will be there. So this pairing of the chromosome, which chromosome? These chromosomes are termed as homologous chromosome. Okay, homologous chromosomes are which one? Which have same size, okay, same genes and different centromeres, you can say. It's not the chromatid. If I'll talk about sister chromatids, they are attached with one centromere having same genes, same alleles. But homologous chromosomes, they are coming from different parents, one from the mother, one from the father, and they have same location of the genes on each structure. Okay, if I'll say the gene for the hair color is located at this particular position in parental paternal chromosome, and in the case of maternal, same location will be there for the same gene. Okay, this is a homologous. So after condensation of chromosome, the pairing of homologous chromosome will take place. So such kind of pair, how many will be formed in a human cell? 23. So this is the one pair of chromosome, one from father, one from mother, and it form a pair. This pairing process is known as synapsis. Okay, this is known as synapsis, pairing of homologous chromosomes. And this pair, ये जो अब जोड़ा बन गया है, this is termed as bivalent. Okay, fine. Now this pairing is facilitated by the formation of proteinaceous structure in between, which is called a synaptonemal complex, which is made up of DNA and protein. So this structure which help these homologous chromosomes to remain together during this stage, this is termed as synaptonemal complex. What we are going to call it? Synaptonemal complex, which will be further disintegrated. Okay, So synaptonemal complex, chromosome from the father, chromosome from the mother, and this complete structure is a bivalent. Okay, now the chromatids will be clearly visible after this particular stage. We'll see how these chromosomes are going to look now. This is again a bivalent which is connected with the synaptonemal complex. Fine. Now this chromosome is having two chromatid, one and this two. This is also having two chromatid, one and two. One is the original, one is the replicated. One is the original, one is the replicated. These two chromatids of the same chromosome, this one and this one, they are termed as sister chromatid. ठीक है, एक ही क्रोमोसोम की chromatids which are connected with one centromere is called sister chromatid. ठीक है, and the chromatid of the homologous pair, ठीक है, ये chromatid, one is this one and one is this one. These are called non sister chromatid. ठीक है, ये दूसरे क्रोमोसोम की chromatids हैं, non sister chromatid. So when the crossing over, which is a very peculiar feature of this particular stage, prophase का जो main change है, that is the crossing over, that is going to take place in between non sister chromatid. ठीक है, जो क्रॉसिंग ओवर होगी वो नॉन सिस्टर क्रोमेटिड्स के बीच में होगी. एंड एज दिस बाइवेलेंट इज प्रेजेंट, ठीक है, सो इसमें आपको कितने क्रोमेटिड्स दिख रहे हैं? 1 2 3 4. ठीक है, 4 क्रोमेटिड्स आर विजिबल, ओके, एट द टाइम ऑफ द पैचेटीन. सो दिस इज कॉल्ड टेट्राड. ठीक है, बाइवेलेंट स्टेज को हम टेट्राड भी बोल सकते हैं बिकॉज 4 क्रोमेटिड्स आर visible over here. Clear? Now when the pairing occur, when this tetrad bivalent, they are clearly visible. The next is crossing over. Crossing over, exchange of the these two were present. ठीक है, ये हमारा synaptonemal complex इनके बीच में ऐसे present था. So how this? We'll just remove this for a while. This way they come close, ऐसे थे ये हमारे, ठीक है, और ये ऐसे एक दूसरे के साथ क्रॉसिंग ओवर के लिए अरेंज हो जाएंगे. सो इन दिस वे क्रॉसिंग ओवर विल टेक प्लेस. क्रॉसिंग ओवर विल टेक प्लेस during which exchange of genetic material will occur in between two non-sister chromatids. ठीक है, ये दूसरे क्रोमोसोम की क्रोमेटिड है, ये दूसरे क्रोमोसोम की क्रोमेटिड है. दोनों के बीच में जहां पे ये एक्स लाइक स्ट्रक्चर बन रहा है, दिस इज टर्म्ड एज chiasma और chiasmata. This X like structure, okay, this cross, this complete one, this is chiasma. Okay, this is formed during the crossing over process. So this is also going to occur during the patchetine stage of the prophase 1, which results into the exchange of material from this to this. And during this process, many enzymes are involved in it. The category of enzymes coming under the recombinase, okay, which will help in breaking the segment of this, attaching that to the another one. Okay, as a result, what will be getting? We'll be getting the recombinates, or we can say we'll be getting the two newly formed chromosomes. When these chiasma are going to break, when this synaptonemal complex is going to get disintegrate, afterwards we'll be able to get these two newly formed chromosomes with exchanged genetic material. This was the point where the chiasma was formed, and this is the segment of this chromosome, this is the segment of this chromosome. These are the recombinants which are responsible for bringing the variations in an organism. ठीक है, यही क्रॉसिंग ओवर है जिसकी हेल्प से हमारे अंदर वेरिएशंस आते हैं. लाइक वी आर dissimilar, we are not exactly same as that of our parent, we are not exactly same as that of our siblings due to the crossing over which take place during meiosis 1. Fine. So this is all about the different changes which are going to occur during prophase. The same we will discuss in our next video lecture. Thank you.